Today we're going to go over topic 6.7 on mutations. Our learning objectives for today are to describe the various types of mutations, explain how changes in the genotype may result in changes in the phenotype, and explain how alterations in DNA sequences contribute to variation that can be subject to natural selection. All right, so let's focus on that our third objective, explain how alterations in DNA sequences contribute to variation that can be subject to natural selection. So when we look at natural selection, which will actually be more in our next unit, Unit 7, we are looking for changes that change how an organism can survive and reproduce. Those two things go together in natural selection. And so in natural selection, we've talked about things like survival of the fittest. It's can that organism survive and reproduce? So when it comes to a mutation is any alteration or change in a DNA sequence. And that mutation, based on through the lens of natural selection, can be either positive, negative, or neutral, meaning that it can have a good effect on that organism's chance of surviving and reproducing, a negative effect, a bad effect, or neutral, no effect at all. So example of something that's a benefit. So there's some plants that have resistance to different infections. That helps that plant to survive and reproduce. This little fly here, if having this extra set of wings makes this fly perhaps faster, now it can escape predators better, and it now survives and reproduces better. Those would be positive things. In bacteria, we have bacteria now that are antibiotic resistant. That's a positive thing for those bacteria. They can survive and reproduce better. Here we have an example of a negative mutation. So here we have some corn plants, and these ones down here to the right have a mutation that prevents them from producing chlorophyll. That's going to be a bad mutation. Plants need chlorophyll for photosynthesis. So this one here, these few down here are not going to survive and reproduce. Perhaps these ones here in the middle might survive, but their reproduction is going to be less. Certainly not as good as the corn plants to the far left. So this is a negative mutation. This plant has a mutation. It can't make chlorophyll. It's not going to survive and reproduce. And then here we have a neutral mutation. So here we're looking at human earlobes, whether they're attached or unattached. And this is one that doesn't have an impact on your ability to survive and reproduce. It's neutral. It's a mutation, it's a change, but as far as natural selection goes, it has no effect on natural selection. This person is not any better at surviving and reproducing compared to people who have unattached earlobes. So let's look at what mutations can affect. So recall that there are two types of cells in your body. You have the gametes or the sex cells. So if the mutation is in a sex cell, that's going to get passed on to the offspring. The parent might not know they have a mutation. Perhaps they got exposed to some radiation, but it only damaged their gametes. Parent could be fine, but then that gets passed on to the future generation, impacting that entire offspring. Depending on where that mutation is, offspring may not survive, but the offspring is capable of passing on that mutation. So in the case of this little fly here, as long as this mutation for 2 wings was also in the gametes, it could pass that mutation could go on to its offspring and potentially lead to a new species over time through natural selection. Somatic cells only affect the individual. So these could impair the function of a cell. but it's not going to get passed on to other generations. So here the gametes were normal. At some point, this diagram does show it happening in the embryo, but it could really be at any point during an organism's life. If a somatic cell gets damaged, they could be affected, but it's not going to get passed on. I like to use the example of skin cancer. You get too many sunburns, you develop skin cancer because of that damaged DNA, but that's not getting passed on to your offspring. All right, so cancer is not going to be passed on. Let's think back now for a second to our fly here. If this mutation happened in a somatic cell, perhaps it was early on when our fly embryo was developing, but it only impacted somatic cells for wings, then that fly mutation would not be passed on to generations, and we would just have one weird fly, but it wouldn't be changing the whole population. So when we get to talking about natural selection later on, mutations have to be inheritable for them to impact a population and possibly cause change or evolution. All right, so some of our causes of mutations.
Students, it's now.
All right, causes of mutations. There can be spontaneous mutations, meaning that there can be an error in DNA replication or just a random error in transcription and making the mRNA. Now, the cell does have mechanisms in place to reduce errors, but it doesn't stop them completely. When it comes to these spontaneous mistakes, errors in replication or transcription are just going to impact one particular gene. Other errors, we can also have errors in cell division, so whether that's mitosis or meiosis. And if we have an error in cell division, that's going to impact several genes because that's going to change the whole chromosome. We'll look at that towards the end of our notes. This diagram here is showing how we could have had a single-celled organism in this case, or at least one that's asexually reproducing, giving identical offspring until we had our spontaneous mutation. Other mutations are going to be caused by factors in the environment that we refer to as mutagens. So mutagen is any external agent that causes a change in DNA. On TV, this tends to be very dramatized as our glowing mutagen here from Ninja Turtles. But in real life, it's far more subtle than having a bottle of ooze poured on you. And so radiation can cause mutations. More commonly though, that's going to be from overexposure to UV light, so from sunlight, too many UV rays, that's skin cancer. Overexposure to X-rays can cause mutations. Now your once a year dental X-ray, it's not going to be enough exposure. But people who work with X-rays will wear lead vests to help protect them, or they'll leave the room. So think about when you're getting your X-ray at the dentist's office, they put a lead vest on you to help reduce that exposure. And the hygienist that's cleaning your teeth leaves the room, moves behind a wall to help reduce their exposure to X-rays. Certain chemicals can alter your DNA. In the case of single-celled things like bacteria, we're looking at high temperatures altering their DNA. We also know now that there are some viruses and as well as some bacteria that can all cause mutations. But in general, a mutagen is anything that causes a change in DNA. Frequently, things like UV light are causing changes just in the somatic cells, that gametes can be altered also. And if the gametes are altered or changed, that's going to get passed on to the offspring. So some of our types of mutations, we have point mutations. So this is a change in a single base. This is a substitution. One thing's being replaced with another. And so this will change just one amino acid, but it changes our entire protein. So if we look down here at our example, here we have the dog bit the cat, substituted for the dog bit the car. So we can still sort of read it. This change here is what we would call a missense. Our codon is coding for the wrong amino acid. And so down here too, we've got another example where a G has been switched for an A. So glycine to serine. Here we've got a change in just one amino acid. This would be a missense. We can also get nonsense. If this had been changed to a stop codon, then we would get a nonsense. Ceasing translation, making a shorter protein. We also have silent. And so this is when the codon changes, but the amino acid stays the same. So recall that our wobble effect, where this third base can change. So if this UUU was changed to a UUC, it could still code for phenylalanine. And so that would be a silent mutation. Here's just another example. So here's our normal sequence and our normal protein. Our missense, we've changed out one amino acid. The glycine is now an alanine. And so this one amino acid is different, and that has now caused a change in the shape of our protein. And with protein, remember that shape is very important to the function. So now the function of this protein is going to be different. There are some similarities. Maybe it still functions a little bit, but not the same. And then here we have a nonsense where our sequence starts off correct, but here we have a letter replaced, giving us a nonsense mutation. And we've now got a totally wrong protein. So as far as the effect this can have on phenotype, so a couple of human diseases caused by substitutions. We've got sickle cell anemia. Here's our normal sequence, GAG, to produce glutamine, change for GTG, and now we get valine, and that changes the shape of our protein. So now we've got the incorrect hemoglobin. So now they're having problems with blood flow and the transport of oxygen. The rest of the sequence is the same, just one amino acid. Another disease is hemochromatosis. This is when your body stores too much iron. And here we've got our change, again, just one base. And both of these, it does happen to be the second base in the nucleotide, or in our codon, the second nucleotide in our codon, our second base. Here we have a G being replaced to a C. That changes our amino acid, cysteine to tyrosine. So now this person's going to have a different phenotype. They have this disease of hemochromatosis where their body is storing too much iron. Our next type is frame shift. This is adding or losing nucleotides, sometimes referred to as insertions or deletions, causing a shift in the mRNA reading frame. And every amino acid after our mutation is going to change, altering our entire protein. And here, our number of bases that we add isn't going to be divisible by 3, because 3 is our codon reading sequence. We do add in 3 or remove 3, then that's going to cause some changes, but it doesn't mess up the whole reading frame. Now here we have an example of a deletion. So our same sentence, the dog bit the cat. Now we have the dog, ick, heck, at, we got nonsense. So So usually with nonsense, things are going to stop shorter. So here we've got the first few amino acids are correct, and then it stops shorter than it should be. Some altered reading frames. So here's another example. We've added in a single nucleotide. And so everything after GLN is different. It's stopping early. So this is our nonsense. Now, if we were to add in extra sets of 3 or delete 3s, we do have a couple of human diseases caused by those insertions and deletions. So one disease is Huntington's disease, and we have some insertions of nucleotides, but it's sets of 3. So we've got our nucleotide CAG, excuse me. This gets inserted multiple times, altering one section of our protein, while the rest of it then stays the same. And this causes some cognitive issues and neurological problems. And Huntington's is, we'll talk more about later. We're going to have a lab on that next week. Another one, cystic fibrosis, caused by a deletion. This time we're deleting the whole codon, so we're taking away 3 of our nucleotides. It's all 3 are together. We're deleting the amino acid phenylalanine, giving us a slightly shorter protein. This one causes lung issues, development of thick mucus. People tend to be very prone to respiratory infections if you have cystic fibrosis. Our last category of mutations then are the chromosomal mutations. These are caused when parts of chromosomes break off or rejoin incorrectly. And this is what's going to happen during cell division. So mitosis, meiosis, our chromosomes can get messed up. We can have deletions where a section goes away. We have duplications, inversions, getting our genes out of order, insertions where part of it's added into a different chromosome, or translocation, pieces switching places. All of these impact whole genes. We've been talking about gene regulation. Chromosomal mutations tend to have a big impact on gene regulation. If we were to have, say, our regulatory genes here, and then the coding gene here, and now they're out of order, or they're in the wrong place, genes aren't going to be turned on and off correctly, causing problems.